Hi, good evening all of you. And here we are continuing our YouTube live streaming program. And this is our second session from biology. And today we are going to discuss about two important chapters. That is nothing but biotechnology principles and processes. And the second one, biotechnology and its applications. And I know that you are all there in the final lap of preparations. Okay, so almost all the topics you covered, I think. And here also, I am not pointing out some important topics, but the main point is nothing but, and we are covering almost all the topics from the two chapters, Biotechnology 1 and Biotechnology 2. Okay, so that's why. And today, one of the most important point you have to notice that is nothing but biotechnology is the concept you are studying on the first time in grade 12. Compared to all other topics in grade 12 units, there is only one unit you are studying on the first time. And nowadays you can see the biotechnology is highly advanced. And in the last year board examination, a question came which is outside the textbook. Actually, the topic is outside. As you all know that what is the question? Actually, it is RT-PCR. And PCR is a topic in biotechnology, you know that. And PCR is a topic in biotechnology principles and process, polymerase chain reaction. And that concept was developed by a scientist named Carey Mullis. And he is known as the father of PCR, the polymerase chain reaction. Polymerase chain reaction can multiply or amplify the DNA segments. So whenever you are giving a DNA segment into the polymerase chain reaction machine, and that is considered as a gene of interest or the gene to be amplified. And the gene, first of all, it undergoes a process called denaturation, and by which the double-stranded DNA is separated into two single-stranded DNA. And that is taking place in very high temperature. More than 90 degrees Celsius temperature is essential for converting a double-stranded DNA into two single-stranded DNA. And the second step is nothing but by reducing the temperature, you are adding two primers are there, and there are 5' to 3' primers are there, and the two primers are responsible for joining with the 3' end of the two single-stranded DNA, and that process is called annealing, and that is done in a low temperature. And after that you can see you need an enzyme. That enzyme which can tolerate high temperature, that is a thermophilic bacterium, and that is what we call the Thermus aquaticus. And the TAC DNA polymerase is used, or TAC DNA polymerase is the enzyme that is extracted from an archaebacteria, a thermophilic bacteria named Thermus aquaticus TAC. And that TAC DNA polymerase is used for extension, and the deoxyribonucleotides are added as substrates, and automatically what happens, the two DNA strands are changing into a double-stranded strands. So simply we can say that amplification of a particular DNA with the help of the enzyme, and the machine is called a PCR machine. And PCR you studied in your grade 12 NCERT textbook. And what about what we can say the RT-PCR? And you can see it is the reverse transcription. When the COVID-19 pandemic period, that the RNA, it's an RNA virus, it's a retrovirus. It is undergoing a reverse transcription. And yesterday we discussed, the last class we discussed, that is nothing but the Teminism. Baltimore and Temin described the concept reverse transcription. That's why you can see the reverse transcription is done in RNA viruses, the retroviruses, and here you can see the RNA to DNA. Okay, so that's what I told you. There may be chance for asking some relevant questions. Okay, so that is why the reason you should be aware about the topics which are most updated. That is nothing but biotechnology. And the biology is highly advanced, especially the field of biotechnology is very much advanced, my dear students. So in the field of agriculture, and as you all know that is what we called the green biotechnology. And in medicine field, it is called a red biotechnology. So there are different applications are there, and such topics we have to discuss along with these two chapters. Okay, so we can directly move into the first chapter, biotechnology principles and processes, my dear students. Okay, so listen, I hope all of you are ready for the class, my dear students. Please be with the part of the class. Okay, thank you. And listen, please, my dear student, the first question. Here you can see the first question, my dear students. What is that? Name the two core techniques that are involved in biotechnology. So, when you are looking at biotechnology, when we are discussing a biotechnological process, all of you look at the board, please. Here you can see when we are discussing the biotechnology into account. What is basically a biotechnology? So that look at the board, my dear student, biotechnology means what? There will be a donor, you can see. So always you can see the gene of interest or the desirable gene is taken from a donor. So for example, the donor is having its own DNA, you can see, and this is the DNA of the donor. And from the DNA of the donor, what we are doing here, and here we are identifying a particular gene of interest. And after identification, we are isolating the DNA segments from the neighboring area. Neighboring area means what? You can see the DNA is located inside the nucleus, and the nucleus contains, there are so many materials are there, including DNA. Along with the DNA, RNA is there, protein is there, lipids are there. So what is that? You already studied. There are some techniques are there for isolation of the genetic material you studied. And how can you remove the cell wall? You studied, if it is a bacteria, you can remove it by lysozyme enzyme. Lysozyme enzyme can digest the cell wall of bacteria. If it is a fungus donor, if the donor is a fungus, you can use chitinase, because chitinase can digest chitin, and chitin is the major component of the cell wall in fungus. And if it is a plant cell, plant cell wall is made up of cellulose, and cellulose is digested by cellulase. Cellulose is digested by cellulase, and that cellulase is responsible for digesting the cell wall of plants. Getting the nuclear contents, and the nuclear contents one by one, the R removed by RNase, and proteins are removed by protease, lipids are removed by lipases. If there is any carbohydrate, they are removed by amylases, and finally, and the DNA is isolated. And that isolated DNA you are pouring very slowly through the side of the test tube. You are pouring the chilled ethanol. The chilled ethanol, when you are pouring the chilled ethanol, automatically what will happen? The DNA is extracted in the form of threads, you can see. And the threads, you can see that thread-like structures are there. The thread-like structures are taken and using a particular process called what? Spooling. S-P-O-O-L-I-N-G, spooling. I think I am okay, right? Any issue? Okay. So welcome back, my dear students. So here, actually, there is a technical issue. That's why there is a block in between. Okay, I think you are okay now. It's okay, right? Okay, and here you can see, my dear students, so we are starting about, or we are discussing about the introduction of biotechnology. And here you can see this is a particular DNA we isolated. And how can we isolate the DNA? We are isolating the DNA using enzymatic method that you already studied under the processes of biotechnology. And in the process of biotechnology, you can see the first one, as you all know that is the enzymatic process for removing the cell wall. If it is a fungus, you can see that is removed by chitinase. If it is bacteria, it is removed by lysozyme. If it is a plant, it is removed by cellulase. And after that, you can see that you are getting the nuclear content only. The nuclear content, you are removing the protein by a protease, removing the RNA by RNase, removing the lipid by lipase. And you are getting, you can see the DNA as an isolated one, and after that, when you are pouring the chilled ethanol, you are getting the DNA as threads, you can see, and that process is called spooling. So that are the topics for removing or isolating the DNA. And after that, here you can see what you are doing. Actually, this DNA you are taking out, and the DNA requires a vector. As you all know that the DNA requires a vector, and using that vector, you are, so vector means what? That may be a different types of vectors you studied. Vectors means are cloning vectors, and the major vectors. What are the vectors you studied? What are the, just remember what are the vectors you studied? One of the most common vectors you studied plasmid, and other one is called cosmid, and you can see BAC, and other one is called what? YAC, and even you can see bacteriophage. Bacteriophage is also a type of vector. So these are the 5 different types of vectors. You can see bacteriophage vector, plasmid vector, cosmid vector, BAC the bacterial artificial chromosome, and YAC the yeast artificial chromosome. BAC and YAC you already studied under Human Genome Project, because in Human Genome Project you can see, and we need a vector. What are the 2 vectors used in Human Genome Project? One is called bacteria, and the one is called what? Yeast are the host, and from the bacterial host, the artificial chromosome, that is called bacterial artificial chromosome, and from the yeast you can see yeast artificial chromosome. Both are used for doing the Human Genome Project. Human Genome Project already you studied under the chapter molecular basis of inheritance. Okay, and here you can see that's about the vector, and using that vector, what will happen? Using the vector, you can see we are doing the transferring into a host. You can see we are transferring into the host, and this is the host by which you can see we are inserting. So the host is having its own DNA. This is the DNA of the host, and what we are doing, my dear students, we are doing, and this is the foreign gene, right? The foreign gene is incorporating with that one. The foreign gene is incorporating with that one means what? The foreign gene, it is also called alien gene, also called exotic gene. The gene is incorporated with the host, and automatically what will happen? That is expressing, and this one is expressing in the host. When it is expressed, automatically what will happen? And that is what we call what? The success of the biotechnology. So basically the biotechnology, we are altering or we are inserting a foreign gene. Altering the DNA of the host. Alter means what? Changing. Either we are altering the DNA of the host or we are inserting a foreign gene. And this is what we call what? Biotechnology. So what is the question here? What is the question here? And what are the 2 core techniques? There will be a chance for asking one more question. What are the 2 core techniques in biotechnology? Look at the board, my dear students. What are the 2 core techniques? The first core technique is nothing but genetic engineering, and the first one is called what? Genetic engineering. What do you mean by genetic engineering? Genetic engineering means what? Altering the chemistry of DNA. So that DNA is very, very important, right? The DNA we are altering. Altering in the sense, either I already told, altering means what? The change in DNA, or you can see we are adding a foreign gene. All together you can see everything in biotechnology, everything is related with the DNA. Everything is related with the gene. That is what we call what? Genetic engineering. And that is one of the most important core technique in biotechnology. That is what we call what? Genetic engineering. And what is the second one, my dear student? The second one is called bioprocess engineering. And there are 2 core techniques are there. One is called genetic engineering, and other one is called what? Bioprocess engineering. What do you mean by bioprocess engineering? Whatever the biotechnological experiments we are doing inside the laboratory, everything should be done in a microbial free environment. A sterile medium is required. Otherwise, what happens? An infection, or what we can say, there is an involvement of any other organism or involvement of any harmful contaminant, all may leads to the what we can say that may affect the success of the process. So that is why the reason you can see whatever the medium we are providing, all should be extremely microbial free and extremely hygienic, and without any infection. So that is why the reason you can see there are two. One is related with the genes and the other one is related with what? The medium. The medium should be microbial free. The medium should be extremely hygienic, my dear student. So these are the two core techniques. These are the two core techniques in biotechnology. So the answer for that question, name the two core techniques, and one is called first one is called what? Genetic engineering, and the second one is called bioprocess engineering. And bioprocess engineering, as you all know, that it is also one more name is there, that is called chemical engineering. But in your textbook, the word is given bioprocess engineering. So you can write the word bioprocess engineering. So genetic engineering and bioprocess engineering are the two key concepts, are the two core techniques in biotechnology. Okay, so moving into the next question. What do you mean by the term restriction endonuclease? As you all know that in biotechnology, there are three different types of tools you have to study in the chapter. One is called the restriction enzyme, and in your textbook a word is there called a restriction enzyme. What do you mean by restriction enzyme? The real word meaning restriction means what? A restricted in a particular area. And generally there are two different types of nucleases are there. No, what are the two different types of nucleases? One is called exonucleases and other one is called endonucleases. What do you mean by exonucleases? Oh, for example, if this is the DNA, if this is a double-stranded DNA, and what do you mean by exonucleases, my dear student? Exonucleases are always cutting at the edges, my dear student. Whenever you are given a DNA, the edges are cut and removed by this particular exonucleases. And what do you mean by endonucleases, my dear student? Endonuclease can recognize a particular recognition. So endonucleases can recognize a particular sequence, and after detecting the sequence, the sugar phosphate backbone. It's very, very important. The DNA we are cutting, not the hydrogen bonds. When we cut the hydrogen bonds, the DNA, the double-stranded DNA is separating into single-stranded DNA. We don't want the single-stranded DNA. We need a particular segment of DNA. Hey, that is what we call the gene of interest, right? And we need that particular gene of interest means what? We have to cut the sugar phosphate backbone. So this is the sugar phosphate backbone. You already studied the structure of DNA given by James Watson and Francis Crick in 1953. And that is the double helical model of DNA. And the double helical model of DNA is given the backbone of the DNA is nothing but the sugar phosphate backbone. So if you need a piece of DNA, we have to cut the sugar phosphate backbone. So the sugar phosphate backbone is cut and removed by the endonucleases are there, and that endonucleases can recognize a particular sequence, and after recognizing, it is cutting that particular area, and this is what we call what endonucleases. And we need endonucleases for biotechnology. Biotechnology, we need endonucleases, and that endonucleases, why that endonucleases are also given the name restriction. What do you mean by the restriction? And for that, I want to explain you one concept, my dear students. Look at the board. In 1963, look at the board, my dear. In 1963, you can see there is a remarkable observation reported in the field of biotechnology. And here you can see, as you all know, that if this is a bacterium is attacked by a bacteriophage. And here you can see this is a bacteriophage. You can see when the bacteriophage infecting means what? It is attaching to the host and transferring only the genetic material. And what is happening actually, here you can see the bacteria, look at the board, here you can see the bacteria developed two technologies. The bacteria developed two techniques to prevent or to restrict the growth of bacteriophage in bacteria. The bacteria developed two techniques. What is the first technique? The first technique is nothing but methylation. The first technique is nothing but methylation, adding methyl group to the DNA of the bacteria itself. That is what we call methylation. Methylation can prevent that infection. And the second one, that is nothing but restriction. Restriction means what? Cutting the DNA. So there are two observations are there. One is the methylation and another one is cutting of DNA. Cutting of DNA is always essential for in biotechnology as an important tool. So that is why the reason, the second one, that enzyme, that there are some special type of enzymes. Were extracted from the bacteria, and that enzymes can cut DNA. Cut DNA means what? What is a word we can use? And they are called nucleases, right? Hey, no, because DNA is a nucleic acid, right? And cutting the DNA means what? Nucleases. And is it cutting on the outer side or in the middle side or in the inner part of the inner part? That's why we are using another term you can see, endonucleases. And you know that enzyme can restrict the growth of growth of that particular bacteriophage. So that's why they use one more word, that is restriction. So one is called restriction, other one is called endo, and the last one is called nuclease. So that's why restriction, endo, nuclease, my dear student. So that's why each one meaning is a nuclease, cutting DNA, endo, the inner part of DNA. Restriction can restrict the growth of bacteriophage in bacteria. Restriction endonuclease, and that is in 1963. So that was a remarkable milestone in the field of biotechnology. And after 5 years, my dear student, in 1968, after 5 years, in 1968, you can see the first REN, the first restriction endonuclease, the first molecular scissors, the first chemical knife. These are the other terms given for restriction endonuclease. Molecular scissors or chemical knife, you can see, and that is what we call what? First restriction endonuclease. What is the first restriction endonuclease discovered in 1968? After 5 years, and they observed that there is an enzyme called HIND2. And this is what we call what? HIND2. What is H? And that enzyme, that restriction endonuclease enzyme, the first restriction endonuclease enzyme was extracted from a bacteria named Haemophilus influenzae, and that was taken from a bacteria named Haemophilus influenzae. And Haemophilus influenzae, that you already studied. How can you put the name? What is the nomenclature of restriction endonuclease? There is a rule. The first letter always should be the first letter of the genus, and the second and third letters always the first and second letter of the species name. That's why Haemophilus influenzae, HIND is the strain, and 2 is the order by which that enzyme is sequenced from the host. And here you can see HIND2, the first enzyme to be isolated, my dear students, and that is the first REN. And you know, after that you can see the most common or what we can say in biotechnology nowadays, we are using the most common REN, that is something but EcoR1, Escherichia coli. E stands for Escherichia, CO stands for coli, and which strain? It is RY13, that also you studied. The R stands for RY13, and that was the first enzyme isolated from Escherichia coli RY13 strain, and that is what we call what? EcoR1. And you know, so far you know more than 900 restriction enzymes are isolated from nearly 230 strains of bacteria. So bacterial strains are taken, and you know, many restriction enzymes are isolated. And here you can see there's an idea about that word, that observation, restriction. So here you can see, so what is the answer for this question? What is the term restriction? The restriction means what? That enzyme can restrict the growth of bacteriophage in bacteria. That's why the term, that's why the reason it is given restriction. Moving into the next question. Differentiate between blunt ends and sticky ends. I think it is compared to an easy question, you know that, but you know, you just listen to the board, my dear. What do you mean by blunt ends? First of all, we can discuss, we are familiar with the sticky ends, right? In your textbook, you can see sticky ends are given. So I already told you, know, one of the most common one, that is what we call EcoR1. The EcoR1 from the RY13 strain of Escherichia coli, and here you can see this is the enzyme, and you know, this each and every restriction enzyme is having a recognition sequence. So you know, EcoR1 cannot cut a DNA at any site. And you know, in the DNA, if there is a recognition sequence, it can attach with that recognition sequence, and it is cutting in a special manner. For example, if this is the DNA, if this is the DNA, if this is 5' to 3', if this is 5' to 3', this is 3' to 5', here you know there should be a sequence. What is that? You already studied GAATTZ. And here you can see this is the palindromic nucleotide sequence you already studied. So that's why GAATTC. And here you can see this is the triple bond, double bond, double bond, double hydrogen bond, double hydrogen bond, triple hydrogen bond, and this is the recognition sequence of eco-R1. If you are asked to write the recognition sequence of eco-R1, you have to draw this diagram 5 prime to 3 prime first, then after the 3 prime to 5 prime. In 5 prime to 3 prime, you have to write GATTC, and 3 prime to 5 prime, you have to write CTTAAG. It is a palindromic nucleotide sequence. You have to study only one palindromic nucleotide sequence, that is nothing but eco-R1. And you know the eco-R1, you should know that in which area it can cut. You can see this is the area. This is the area between G and A. This is the area between G and A. So when you are cutting in between G and A, when you are cutting in between G and A, you can see a sticky end is produced. And how is sticky end is produced, my dear students? Here you can see, when we are looking the diagram, here you know only up to G. And here you can see, when you are looking here, you can see here G, triple bond, the C will be there, and T will be there, T will be there, A will be there, A will be there. And this is one sticky end. Okay, and what will happen, because you know, and this is the area where the restriction in the nucleus can cut the sugar phosphate backbone, and here this is the area where the enzyme can cut the sugar phosphate backbone. So that's why the remaining part is removed. So what is the remaining part? In this side you can draw like this. Here G, A, T, T, C. C means what? Triple bond G, and here this is what we call what? The two sticky ends. So here you can see this is the eco-R1. The eco-R1 can make two sticky ends. Two sticky ends you can see, and it is joined by the molecular glue. It is nothing but the DNA ligase. And this is the biotechnology. This is the sticky end means what? What do you mean by blunt ends, my dear students? And you know, so that's why the blunt ends, you know, there is no example given in your textbook, but you have to study. There is one example, is there? Eco-R5. Easy to study that one. So this is eco-R1 means what? R means what? RY13. Here also RY13. Then why it is 1? 1 means what? The first enzyme isolated. And that is 5 means what? The fifth enzyme isolated. Okay, the fifth enzyme isolated from RY13 from Escherichia coli. And here you can see what is it. Just, no need to study in detail like this. So just giving the idea. So here you can see it is 5 prime to, look at the board, my dear student, 5 prime to 3 prime. And this is 3 prime to 5 prime. And what is that? Here you can see GAT, my dear students, and ATC. GAT and ATC. So GAT. So here you can see GAT, ATC. So here you can see GAT, ATC. And here the triple bond, double bond, double bond, double bond, double bond. Here you can see triple bond. This is GAT, ATC. Don't confuse with GAT, ATC with GAT, ATC. GAT, ATC is not in the textbook because it is eco-R5 and it is for explaining the blunt ends, my dear student. Blunt ends means what? And where is that? Here you can see, when we are looking this diagram, you can see that here you can see the enzyme can make a cut in between A and T. And here you can see here T and A, and here also they are making a cut over here, my dear students. And here compared to the first diagram, you know, this is totally different, right? And here you can see cutting between, and the edge is cut, and here the middle part is cut. So you know, in eco-R5, here you can see GAT, ATC. And what will be the result, my dear student? What will be the result? What will be the result? How can I draw the diagram? You can see. So you know, when I draw the diagram, look at the board, please. When I draw the diagram, here you can see 5 prime, here you can see 3 prime, here GAT will be there, and here ATC will be there, triple bond, double bond, double bond. Here another piece you can see, this is ATC, and you know TAG, and here triple bond, double bond, double bond. And you know what is this end called what? And you know, you can look at the two pictures. Here you can see this is what we call the sticky end, and this is what we call what? Blunt ends. And the blunt end example is no there, so that is called blunt end and sticky end. And you know, in biotechnology, the most common one as well as the most what we can say suitable one is nothing but the sticky ends. That's why only sticky ends are explained as well as given in your textbook. But at the same time, you keep your mind, along with the sticky ends, blunt ends are also there. Okay, I think it is clear, and this is the difference between sticky ends and blunt ends. Blunt, sticky. Okay, moving into the next one. Sieving effect is observed in agarose gel electrophoresis. Justify. Agarose gel electrophoresis, as you all know that what is agarose gel electrophoresis, is a technique by which that is used for separating the DNA segments according to their size. And my dear students, you know, I don't know how many of you are remembering the term southern blotting. This is, you are not supposed to confuse southern blotting with agarose gel electrophoresis. Agarose gel electrophoresis is for separating the DNA segments based on their size, and southern blotting means what? We are transferring the DNA segments, and that is also used for separating the DNA segments, but that is blotting technique. In DNA fingerprinting, we discuss that topic. And here you can see the agarose gel electrophoresis. You are very familiar with the diagram. In agarose gel electrophoresis, you can see very simple. Here you can see there are some wells are there, and here you can see the opposite side. You can see this is the positively charged area. You can see this is the anode. You can see, and here in between you can see the gel. Agarose gel, my dear students. Here in this area we are putting agarose gel, and agarose gel means what? It is taken from a seaweed called agar, a red algae. You studied, no, rhodophysae. Grade 11 you studied, no, the algae. There are 3 different types of algae you studied, right? What are the 3 different types of algae you studied? Not related with the grade 12, but you know that is important for your NEET examination. Okay, so the algae, there are one is called chlorophysae, the green algae, phaeophysae, the brown algae, and the rhodophysae, the red algae. And the red algae, you can see there are different types of red algae, and you know the red algae can produce the agarose gel, my dear students. Here you can see the agarose gel. You know, it is just like a sieve, net-like, mesh-like structure, my dear student, and it is having a net-like structure. This is the agarose gel. So that's why what will happen, when you are keeping the DNA here, when you are keeping the DNA fragments here, and this is the DNA fragments we are filling in the wells. You can see, you already studied DNA is highly negatively charged. DNA is highly negatively charged, and DNA is highly acidic. In the packaging of DNA you studied, the DNA is packed using positively charged histone protein and what we can say, the alkaline material, the histone protein. So here you can see whenever we are keeping the DNA fragments here, you can see whenever it is connected with so the electrophoresis, right? Whenever it is connected, my dear students, automatically what will happen? The DNA is having a tendency to attract towards the opposite pole. So here positive means what? DNA is negatively charged. Negatively charged materials are moving towards the positively charged area. So when it is passing through the agarose gel electrophoresis, what will happen? Just like a net means what? Small fragments, very important. The small fragments can move faster, my dear student, because net-like structure, right? Large fragments, that will take time, or that can move very slowly. So that's why you can see, look at the board, my dear student. Here you can see the larger fragments are always close to the wells. And what about the smaller one, my dear student? The smaller one, the smaller ones are moving away from the wells, and compared to the first one, the second one is smaller, right? So smaller one is moving away from the well, and again you can see the smallest one is again moving away from the well. So that's why the DNA segments are separated based on their size. Because through the net, when we are passing a material, you can see the smaller fragments can move faster, and while you can see the larger fragments move slow in nature. So that is why the reason you can see the larger fragments are always located close to the well, and smaller fragments are moving faster, my dear students. So in this point, you can say that, and when it is passing through a gel means what? Agarose gel means what? Acting as a sieving effect. And actually a sieving is just like a net-like, just like a mesh-like structure. That's why it is called what? A sieving effect. As you all know that after that, you already studied which stain you are using, my dear student. You are using a stain called ETBR. What is ETBR? ET stands for ethidium, BR bromide. ETBR, that is ethidium bromide, and ethidium bromide and ultraviolet rays. After adding the stain ethidium bromide, we are passing the ultraviolet, and you can see brightly orange colored DNA bands you can see in the agarose gel electrophoresis plate. And after that, we are doing one process. What is that process? That also you studied. What is that process? We are cutting the DNA along with the gel. We are cutting the DNA along with the gel. And that process also, there is chance for asking a question. That is what we call what? Elution. Elution is the technique by which we are taking the DNA along with the gel. That is what we call what? Elution. And here that is sieving effect. And whenever there is a question, sieving effect, if it is a 1 mark question, if it is a 2 marks question, 1 mark is always MCQ, right? If it is a 2 marks question, you can write in words. But if it is a 3 marks question, I already told, no, my dear students, if it is a 3 marks question or 5 marks question, wherever it is necessary, it is your responsibility to draw the diagram. And agarose gel electrophoresis diagram is comparatively a very easy diagram. So if it is required for a 3 marks question, you should Draw the diagram in the board examination paper. Moving into the next question. What are the major recognition? What are the major recognition sequences in PBR322? So you know that, my dear students, when we are discussing the PBR322, so you know it is not a natural plasmid. You know that PBR322 is always a artificial one, a synthetic one. So here you can see this is the plasmid. You can see the plasmid there are. Is it a single stranded or double stranded? As you all know that plasmid is always double stranded DNA. Is it chromosomal or extra chromosomal? It is extra chromosomal. I will repeat one more time. Is it single stranded or double stranded? It is double stranded. Is it linear or circular? It is circular. Is it chromosomal or extra chromosomal? It is extra chromosomal. And it is also having one more property. It is self-replicating, autonomously replicating extra chromosomal, double-stranded, circular DNA that is extracted from the bacteria is called what? You know, plasmid, you know that. And that plasmid you can see, and it is double-stranded, so that's why you have to draw one more line, and this is what we call what? The double-stranded extra chromosomal circular DNA. Okay, and here you can see what is that? And all the DNA or all the plasmid, you know, it is that natural plasmid itself. Two areas are quite common, and this is what we call what? ORI, the origin of replication, and just near the ORI you can see the ROP site. ROP means what? ROP, repressor of the primer or replication. The copy number is determined by the ROP site, which is located close to ORI. And this is a natural plasmid taken from E. coli. But you know, the natural plasmid we cannot utilize in biotechnology, because in biotechnology we want to separate the recombinants and non-recombinants, as well as we have to separate transformants and non-transformants. So the recombinants and non-recombinants and transformants and non-transformants both are separated using some selectable markers. What is the word I use? Selectable markers. And that selectable markers are surely, as you all know, that antibiotic resistant genes are there. So that is why the reason here we are inserting an antibiotic resistant gene. Here we are inserting an antibiotic resistant gene. What is that antibiotic resistant gene, my dear student? It is what we call TETR. What is that? Tetracycline resistant gene. Okay, so you know that gene can resist the tetracycline antibiotic. And what is this one, my dear student? Another antibiotic we are adding in the opposite side. It is AMPR. It is ampicillin resistant gene. So there are two genes are added, my dear student. One is called what? TET-R, other one is called AMPR. TET-R, the tetracycline resistant gene, and AMPR, the ampicillin resistant gene. These are the two selectable markers, and the two selectable markers, one for selecting the recombinants from the non-recombinants, other one for selecting the transformants from non-transformants. And here you can see this is the basic structure of a plasmid. But the question is not ask you to draw the basic structure. The question is what are the major recognition sequences? Recognition sequence. So far, I didn't mention anything about recognition sequences. And what do we mean by recognition sequence? You know, whenever an enzyme, whenever a restriction endonuclease, whenever an enzyme restriction endonuclease, the restriction endonuclease must have a recognition sequence. Then only that area is cut and removed. So that is why then only we can do recombinant DNA. What is recombinant DNA? Recombinant DNA is a combination of two DNA. Maybe one is from the donor or the foreign DNA, and other one from the plasmid or vector DNA. Both are combined using a molecular glue called DNA ligase, and that is what we call what? The recombinant DNA. The recombinant DNA, the first recombinant DNA. How many of you can remember the first recombinant DNA? The first recombinant DNA was developed by two scientists called Stanley Cohen and Herbert Boyer, my dear students. Stanley Cohen and Herbert Boyer developed that first recombinant DNA, and in 1970s. And what was their experiment, my dear student? They took a plasmid from Salmonella typhimurium bacterium. They took a plasmid from the Salmonella typhimurium bacterium, and they insert an antibiotic resistant gene, and finally that is transferred into the E. coli bacteria. And after that, the E. coli bacteria is getting the antibiotic resistance. That is what we call the first rDNA technology. So here you can see AMPR and TETR, and this is what we call what the basic one. And what are the recognition sequence? That always a confusing topic for the students in grade 12, because you know, how can we study that 8 recognition sequence? And how can we study, my dear students? Here you look at the board, you know what are they? What are the 7? One by one you look at the board, my dear students. Here you can see what is the first one. Here you can see this is the first one. It is called what? PST1. Actually, that is from a Providentia Stewarti. No need to study the full form. At least you have to study PST1. And here you can see a PVU1. PVU1, my dear student. What is PVU1? Proteus vulgaris, you can see. So PST1 and PVU1 is there in the ampicillin resistant gene, and here you can see one is there here, EcoR1. EcoR1 is there in this area, and you know CLA1 you can see, and here you can see HIND3. EcoR1 here, CLA1 here, HIND3 here, and you know that three recognition sequences are there between ampicillin and tetracycline. And after that, here you can see the BAMH1, and here you can see SAL1, and here in the ROP you can see a PVU2. As you all know that it is a little bit difficult to remember all the 8 recognition sequence, my dear student, and PST1, PVU1, EcoR1, CLA1, HIND3, BAMH1, SAL1, PVU2. How can you remember all these things? And we all know that what is that? That we can do only with the help of connecting it with a cricket match, my dear students. How can you connect this one? How can you connect the plasmid with the cricket match? And not the recent cricket match, you can see 10 years back or something, you can see the Indian cricket were ruled by 2 eminent openers, my dear students. Do you have any idea about the openers? Rule the Indian cricket, that's nothing but Sachin Tendulkar and Virendra Sehwag. And here you can see, and here you can see, just giving a clue only that you can use it or you can reject it. That's not a matter. Here you can see the first one. What is ST? ST, you can see the PST, you can see ST is the Sachin Tendulkar, and VU the Viru, and you can see there are 2 openers, and that's why PST1 and PVU1, they open the innings. And you know, in the open the innings means what? Both are there in the ampicillin region, PST1 and PVU1, and they open the innings. And you know, when they open the innings, as you all know that they will play, their cricket is very awesome cricket, right? So that's why they are playing in, and the first economy rate, the best economy rate they are playing, and that is the advantage of Indian cricket. You know that Sachin Tendulkar, we are all believing in Sachin, right? So that's why Sachin and Viru together, they played well. That's why EcoR1, the best economy rate. And when they are playing in best economy rate, what will happen? The audience will clap, right? Automatically the audience will clap, because that's a beautiful match, right? And that's why the audience clapped, my dear student. And which audience? It is nothing but the Indian audience, right? Because you know the HIND3 means for the Indian audience, because you know our Sachin Tendulkar, right? Our Virendra Sehwag, right? The HIND3. And after that you can see, and after that the match is going on very well. So that's why there is a commentary, and most of the commentaries are in Hindi, right? And bahut acha match hai. Actually, a Hindi commentary is going on. Bahut acha match hai, my dear student. And that's why the BAMH is there in tetracycline. Why it is a bahut acha match hai? The reason is there because Sachin scored 100 at the same time. And you can see at the same time the Sachin scored 100 and the PV, the Viru scored 100. So, this is, you just connect it with a cricket match. There is no other option to study everything in the correct order. Repeat one more time. Sachin Tendulkar, Viru opened the innings and they played well in well economy rate. And audience clapped, the Indian audience clapped, bahut acha match hai, the commentary, and after Sachin scored 100 and the Viru scored 100. Okay, so that is the only method to remember all the 8 recognition sequence in PBR322. What is this BR, my dear students? What is this BR? And the BR, that is also not given in the textbook, my dear student. The B stands for Bolivar, my dear student. B stands for Bolivar and R stands for Rodriguez. And these are the 2 scientists. Bolivar and Rodriguez are the 2 scientists. They developed and these 2 eminent, you can see the personalities, they developed the first artificial plasmid. That is what we call PBR322, Bolivar and Rodriguez. Okay, so that's why the question is, what are the major recognition sequence? And there are 8 recognition sequence I represented here in blue in color, starting from PST1 and that ends in PVU2. I hope it is clear. Moving into the next question. Why Agrobacterium tumefaciens? Why Agrobacterium tumefaciens is considered as the natural genetic engineer? There is chance for asking that question because it's a repeated question, my dear student. Why Agrobacterium tumefaciens? As you all know that Agrobacterium, as the word indicates, it is related with the plants, right? Agrobacterium. And Agrobacterium tumefaciens is a special type of bacterium, and the speciality of this bacterium, it is a pathogen. It is a pathogen. It is a pathogen to the dicot plants, you can see. For example, and the Agrobacterium tumefaciens, as you all know that there is a special type of plasmid is there, and that plasmid is called what? TI plasmid, my dear student. And what is that plasmid called? What? TI plasmid, the tumor-inducing plasmid. The TI plasmid is called the tumor-inducing plasmid, my dear student. And you know how it can. Actually, this figure is not there. In your textbook, the explanation is there. TI plasmid, the tumor-inducing plasmid. And the tumor-inducing plasmid, you can see there is one region is there. Look at the board, my dear student. This is the region, a special region I marked over here. And that is what we call the tDNA region. And what are they? And what is the name of the plasmid? You can see this TI plasmid, the tumor-inducing plasmid. And the tumor-inducing plasmid is having a region I marked here and a shaded region. And this is called the tDNA, the transfer DNA region. The transfer DNA region can infect the plant, can infect the dicot plant and can cause the tumors, my dear student. Tumor-inducing, right? Transfer DNA can cause that particular tumor disease. So that's why automatically what will happen, my dear students? What we are doing? We are doing a method. What is that? We are inserting a foreign gene here. Where we are inserting? We are inserting a foreign gene in tDNA. So when you are inserting a foreign gene in tDNA, it can pass or it can transfer into the host, but it cannot cause tumor because the tumor causing area. And this is what we call the left border. You can see this is the right border, this is the left border. In between, you can see there are different types of hormones are there, phytohormones are there, auxin, cytokinin, gibberellin, different types of phytohormone producing what we can say genes are there in that area, my dear student. Phytohormones are plant hormones. So that's why what will happen? Here you can see the tDNA region is transferred, but they are insertionally inactivated. When you are inserting a foreign gene, into a plasmid DNA, that particular region is inactivated. Insertion, inactivation, and that process is called insertional inactivation. And due to insertional inactivation, what happened, my dear student? And it cannot cause tumor. So that's why naturally the Agrobacterium tumefaciens can naturally enter into the host plant. Naturally, it can enter into the host plant by carrying a foreign gene. What is the word I use? The Agrobacterium tumefacien bacteria can naturally enter into the host plant, enter into the dicot plant by carrying a foreign gene. We can use that word. We can use the word natural, right? Naturally, it is carrying. And what is genetic engineer? Why they're called genetic engineer? It can carry a gene, right? It can transfer a gene, right? That is what we call biotechnology. That is what we call the genetic engineering, right? So that is why the reason Agrobacterium is called the natural genetic engineer. Moving into the next question, what is the best method? What is the best method for transformation in plant cell? And you know that when we are taking a host into account, my dear student, when you're taking the host into account, a host is competent only when a foreign DNA enters into it. Host may be a plant, host may be an animal, host may be a microbe, whatever the nature of the organism. How can you make a host competent? Or how can you transform? How can you perform a process called a transformation? How can you transfer the foreign DNA? How can you transfer the alien DNA? How can you transfer the exotic DNA into the host? That is always an important task in front of the biotechnologists. And that transformation is done by 4 different technologies. And only 4 are given in your textbook, but it is not 4. There are more than 10 different transformation procedures are there, my dear student. But you have to study, we have to study only 4. And the first one is nothing but the heat to shock method. You know that. What is heat to shock method? A shock, not electric shock, heat to shock at 42 degrees Celsius. That heat to shock is given in a particular medium, and that medium contains calcium ions, the medium contains the recombinant DNA, the medium contains the host. All together, first of all, we are incubating in ice. The very next one, we are increasing the temperature at 42 degrees, only very short period of time. The very next moment, we are again incubating in ice. During that heat to shock, the recombinant DNA enters into the host, called heat shock method. The second one, that is what we call micro injection. As you all know that micro-injection means what? Using a specialized microscopic syringe-like structure, we are taking the recombinant DNA and injecting that particular recombinant DNA into the host cell directly. That is called micro-injection. And the question here is, which is the best method for plants? You can see the best method for plants. The third one, that is nothing but the biolistics. In your textbook, it is given. Biolistics is also called gene gun, my dear student. Chance for asking that question. Because what is gene gun? Gun is having bullets, right? Here also the biolistics is having, or biolistics machine is having special type of microparticles just like bullets. And the microparticles are made up of gold or tungsten. The microparticles are made up of gold or tungsten. That microparticles are coated with the DNA. The microparticles are coated with recombinant DNA. And using a special pressure, using the helium ion, you can see they are forcefully bombarded. That's the same word used in your textbook. And you can see forcefully bombarded into the host cell, my dear student. And using that force, you can see the recombinant DNA is entering into the host cell, entering into the plant cell. And you know the microparticles are prevented by a screen in front of that host. So that is what we call what biolistics. And the fourth one, that is disarmed pathogen. Disarmed pathogen, we already discussed. What is that? Taking a pathogen and making it inactive by inserting the foreign gene, insertional inactivation. And we can convert a pathogen into disarmed pathogen. These are the four methods for making a host competent, or we can say that these are the four methods for transformation. Moving into the next question. Enzyme used in PCR. In the beginning of the class, I already mentioned about PCR, the polymerase chain reaction. And the PCR, the process PCR, the concept was developed by a scientist named Kary Mullis, my dear students. A scientist named Kary Mullis. The name of the scientist is not there, but when you're writing something extra in the board examination paper, that will be good. So that's why the reason the PCR, the polymerase chain reaction, and was developed by a scientist named Kary Mullis. And you know that what the contribution from who proposed the term a DNA fingerprinting, my dear student, that is Alec Jeffreys. It is given in the textbook. Alec Jeffreys proposed the concept DNA fingerprinting or DNA profiling. The father of DNA fingerprinting, that's nothing but Alec Jeffreys. This is Kary Mullis. And here you can see in PCR the three steps also I mentioned in the beginning of the class, that is denaturation, annealing, and extension. In the extension procedure, we need an enzyme, right? And that enzyme is called what? DNA polymerase. And it is not the normal DNA polymerase, and the DNA polymerase is taken from a special type of bacteria found in extremely harsh climatic conditions. The bacteria which are living in extremely harsh climatic conditions are called Archaebacteria. The bacteria which are living in normal climatic conditions are called Eubacteria. You know, the kingdom Monera is divided into two subdivisions, that is called Archaebacteria and Eubacteria. The Carl Woese was the scientist who classified kingdom Monera into Archaebacteria and Eubacteria. In the chapter Microbes in Human Welfare you studied, and when we are discussing the topic microbes in biogas production, there is a bacteria named methanogens, methanobacterium, that is also coming under this particular category, Archaebacteria. So here also one Archaebacteria, it is called a Thermus aquaticus. What is the full form? Thermus, T-A-Q. T stands for Thermus and A-Q stands for aquaticus. So this is the bacteria, Thermus aquaticus bacteria, and which are living in high temperature condition. That is thermophilic bacteria. And that bacteria, from the bacteria, we are taking an enzyme called Taq DNA polymerase. That Taq DNA polymerase is used in PCR. That is a question. Enzyme used in PCR, that is Taq DNA polymerase. Here you can see list out the major 6 units in a stirred tank bioreactors. As you all know that in your textbook, you can see a topic is there after explaining the process of biotechnology. As we all know that the biotechnology means what? This is the recombinant DNA. Recombinant DNA is making a protein, right? As you all know that recombinant DNA can make an RNA. That RNA can make a protein means what? That protein is called a recombinant protein. And nowadays you can see most of the drugs, everything, all are the basic principle behind the proteins which are not producing any side effects. and they are targeting only that particular specific pathogen, and there are so many advantages are there in recombinant drugs and recombinant medicines. Nearly 30 recombinant drugs are marketed worldwide. Out of the 30 recombinant drugs, 12 of them are marketed in India. It is given in the chapter Biotechnology and its applications. And here you can see the recombinant protein. Here you can see the recombinant proteins. Always we need in large scale. In our laboratory, we can make a maximum of 10 liters or 100 liters, but we need thousands of liters because you can see the human population is rocketing day by day. So that's why you can see the demand for the drugs are also increasing, right? So automatically what will happen? And we have to make a huge quantity of, so that's why reactors, just like electricity is produced by nuclear power reactors, right? And here you can see the large quantity of recombinant proteins are produced by powerful bioreactors. And that bioreactors, you can see the bioreactors, and you know it is looking like a tank, you know that. And here you can see it is looking like a tank. Here you can see this is a tank-like structure, and here you can see all the contents are added into that particular machine, bioreactors. And you know in the bioreactors there are some structures are there for mixing the contents. So altogether you can see the bioreactors. There are two different types of bioreactors you studied. No, so stirred means what? Always stirring, always agitating. So that's why the content should be thoroughly mixed. You know that. So when the stirred tank bioreactor is mixing the contents, my dear students, you know automatically you can see they are combining with each other and they are making the recombinant protein. We can collect 1,000 liters of recombinant proteins. We can collect from the same machine. And there are two different types of stirred tank bioreactors are described in our textbook. One is called simple stirred tank bioreactors, and other one is called what? Sparged stirred tank bioreactors. So stirred tank bioreactors are of two types: simple stirred tank bioreactor, sparged stirred tank bioreactor. These are the two types of bioreactors. And here you can see sparged tank bioreactors or sparged stirred tank bioreactor is always in contact with air bubbles. And that bubbles are there, you can see, so more surface area, and that is called sparged. The bubbles are sparged, my dear students. And here you can see the recombinant protein. Here, what is the question actually? List out the major 6 units. So what are the 6 units one by one? I will explain one by one. The first one, there should be an agitator. There should be an agitator or stirrer to mix all the contents. And the second one, after agitating, what you can see, and when we are agitating, these are all living components, right? Enzymes are added, and you can see the nutrient medium is added. And when we are agitating forcefully, automatically you can see foam will be produced. So that's why there should be a foam control system. So, you can connect it. When we are agitating, there is chance for foam, FOAM, and that foam should be well controlled, right? That's why foam control system. And after the foam control, it is a living structure, right? So that's why you can see oxygen delivery pump is important, my dear student. Oxygen delivery pump, because it's a living structure, right? Oxygen delivery pump. And the fourth one, as you all know that. So one more time, agitator, foam control system, oxygen delivery pump. And after that, you can see a pH control unit is very important. pH control system. What is pH? You can see a pH 1 to 14 you study, 7 is neutral. And in some reaction, in some chemical reaction, pH less than 7. In some reaction, more than 7. In some reaction, it is 7. That depends upon the nature of that chemical reaction. We have to provide the proper pH. When there is a change in pH, you already studied in the last year in chapter biomolecules. Whenever we are moving the pH from the optimum pH, the chemical reaction rate is declining. You study, temperature and pH are having almost similar value, and a reaction is having an optimum temperature, and a reaction is having an optimum pH. When you decrease the temperature, when you decrease the pH, when you increase the temperature, when you increase the temperature and pH, automatically what will happen? That will reflect in the rate of chemical reaction, reflect in the sense decreasing. So you have to provide optimum temperature, you have to provide optimum pH, that is a temperature control system. And these are the 5 major one. What is the last one, my dear student? And we are doing a biotechnological experiments, right? So that's why after 1,000 liters, if we make a product and if the product is not suitable for our purpose, what will happen? The whole effort will be in vain. So that is why the reason. And at proper interval there should be a sampling port, my dear student. What is sampling port? And after a particular time the process is stopped, a sample is taken and tested. Only after testing the sample only the process is continued, my dear student. And that is why there is a port, PORT, there is a port is required for taking the sample, that is called a sampling port. And these are the 6 units in stirred tank bioreactors. Okay, and one more question from the chapter Biotechnology Principles and Processes. What is the question? And here, what do you mean by the term downstream processing, my dear students? There is one question that is called a downstream processing. And what is downstream processing? Here you can see, and you are making a recombinant protein. You are making a recombinant protein. And that recombinant protein, you are not supposed to, market in the next day. When you are making a recombinant protein, the very next day, you cannot market it out. And before marketing, this is the marketing, my dear students. And before marketing, you should do some special techniques. You can see, for example, the first step is nothing but what you should do separation techniques. Okay, and the recombinant protein with some other material, all other material should be removed. Only the recombinant protein, that is called a separation technique. And you have to use the purification technique, my dear student. And if there is any impurities are there, that impurities must be completely removed, and that is called purification technique. And after that, you can see proper preservation is required, right? And preservatives are very, very important. Proper preservatives are added for long lasting that particular product. And there is a quality control techniques. And quality control techniques means what? And each and every product before marketing, we should do the quality control technique. And even the clinical trials are also done. What is clinical trial? If it is a recombinant drug, before selling in the market, before selling in the market itself, that should be clinically tested, okay? Otherwise, what will happen? For example, you are making a human insulin artificially. That should have the capacity to reduce the blood glucose level. That should not produce any side effects in the person who is using. And altogether, clinical trials also must be done. And you can see in between these two green, and I wrote so many red color lines. What is that? Here you can see, before marketing a recombinant protein, we should do series of steps. The series of steps what I marked over here, and that is what we call what downstream processing. And downstream process is different in different recombinant products. Okay, so that's about the topic biotechnology, the principles and processes, my dear students. So biotechnology and principles and process, and when we are discussing that topic, we covered almost all the topics, my dear student. I hope it is clear. We can go for a very, very short break, and after that we can discuss the topic biotechnology and its applications. Thank you. Hi. Okay, so we can continue. My dear students, I think all of you are there. Okay, we can continue, my dear students. You know, this is biotechnology, principles and processes we completed. And we are moving into the second half of the chapter. Okay, so there are two, biotechnology 1 and 2. And this is the second one, that is biotechnology and its applications. Okay, so biotechnology and its applications, you know, when we are discussing that topic, before moving into the topic, you should know that what are the applications, my dear students? What are the applications of biotechnology? Look at the board, please. When we are discussing biotechnology into account, biotechnology is having lot of different applications, my dear students. Different types of different applications are given by the biotechnology, because recently the biotechnology is having several advancements. So look at the board here. Here you can see when we are looking this one. First of all, you know, there is a branch of biotechnology called green biotechnology. What is green biotechnology? Green biotechnology is the biotechnology that is everything related with agriculture. Green biotechnology is the biotechnology that is everything related with agriculture. And what is red biotechnology? What is red biotechnology, my dear student? Red biotechnology is everything related with medicine. So everything related with medicine, everything related with drugs, and that is what we call what? The red biotechnology. So the green biotechnology and red biotechnology are the two key concepts we have to study under this chapter. Okay, so there are so many other brown biotechnology, yellow biotechnology, white biotechnology, even the golden biotechnology is also there, my dear student. And golden biotechnology means what? Biotechnology, everything related with the bioinformatics. And the biotechnology is everything related with the bioinformatics. You know, everything is related with the computational data, the highly advanced field of biology, that is bioinformatics. And that is what we call what? The golden biotechnology. Even you can see the dark biotechnology is also there, my dear student. What is dark biotechnology? And you know what is dark biotechnology? And you know, biotechnology is misused, my dear student. Biotechnology is misused by several multinational companies to make some harmful products, you can see. And you know, that is called a dark biotechnology. And there is a term you are very familiar with, bioterrorism. It's not the normal terrorism, bioterrorism. Actually, there are different strains of bacteria, there are different strains of viruses are spreading all over the world. And that is called bioterrorism, my dear students. So to prevent bioterrorism, there should be some organizing committee to address the validity and ethical issues, right? You already studied, you know, when we are discussing the Human Genome Project, we studied one important concept, addressing LC, ELSI, ethical, legal and social issues. And when we are introducing a foreign DNA into a host, you can see it is our responsibility to address the validity of the research, to address the ethics of the people, my dear students. Ethics means what? Legally and religious issues, social issues, ethical issues, all are very important. Because you know, biotechnology, you are making a foreign material means what? That is a threat to the indigenous varieties. So that's why we are handling, or we should handle, the biotechnology in utmost care, my dear students. And here you can see there are different branch of biotechnology. Here you can see the first question. What is the first question? Nutritional enhancement. As you all know that when we are discussing the biotechnology and its application chapter, and it is given the first one is nothing but, you can see there are three different types of farming is there. There are three different types of agriculture was reported in India. And the first one, as you all know, that agrochemical based agriculture. Agrochemical based agriculture means what? Both fertilizers and pesticides are used, and that is for tripling the production of crops, you can see. The green revolution. Green revolution is one of the best example for agrochemical based agriculture. Wheat is the best crop adopted during that particular period, you can see. Okay, and Dr. M.S. Swaminathan considers the father of Indian green revolution, my dear students. Okay, so green revolution, and there are so many disadvantages are also there, because you know, when we are using the agrochemicals, there are a lot of environmental issues in the form of pollution. Pollution in the sense, you can see the environmental pollution in the sense, you know, water pollution, soil pollution, air pollution, and there are different types of pollutions are also reported along with the green revolution. And after that, after that green revolution, the new type, or the second type of farming came, that is nothing but the organic farming. And organic farming is nothing but, you can see, without using any inorganic chemical, without using any pesticide, without using any fertilizers, we are farming, my dear student. What is the advantage? There is no side effects, there is no pollution. But one disadvantage is also there. What is that? The yield is always less. So, both agrochemical based agriculture and organic farming, both two limitations are there. The first one with full of environmental issues, and the second one regarding the quantity of the yield. And these two problems are solved by, these two problems or these two demerits are solved by the third one that we are going to discuss today, that is genetically engineered crop-based agriculture. Genetically engineered crop-based agriculture, or simply we can say that GMO, genetically modified organism. So the genetically modified organism, the genetically modified crops, you can see the genetically modified crop. There are different types of genetically modified crops you studied. Genetically modified crops are having some advantages are there. What is that? They are tailor-made plants are there, and they can resist abiotic stress. You can see they can increase the mineral usage, they can decrease the post-harvested loss, they can increase the nutritional value of the food, and there are different advantages are there for genetically modified organism. And here you can see, and from the from the advantages of genetically modified crop, and we put the first question here: what is the nutritional enhancement, my dear student? What is the nutritional enhancements? As you all know that the example is nothing but the golden rice, my dear student, the golden rice. And a Switzerland company developed a special biotechnological product named golden rice. It is not in the normal color of light, it's golden in color, and you can see a beta carotene gene is there, and the beta carotene gene is the foreign gene. And beta carotene gene, my dear student, and beta carotene gene is providing the golden color. At the same time, the beta carotene gene is acting as the precursor. Beta carotene gene acting as the precursor for vitamin A. So that's why generally you can see when you are eating a normal rice, when you are taking a normal rice, you are getting only carbohydrates, right? But when you are taking the golden rice, the biotechnological product, you can see the nutritional enhancement you are getting an additional vitamin. What is additional vitamin? That is nothing but vitamin A, which is good for your vision, VISION, vision. There are so many people in India are suffered due to night blindness or vitamin A deficiency disorders, my dear students. And they are not getting the proper vitamin A supplements. Vitamin A is present in, you can see, the leafy vegetables, fruits and vegetables, my dear students. So that's why, most of the people cannot afford that type of fruits and vegetables. So that's why they are lacking the vitamin A, that is leading into a condition called a night blindness. What is the solution for avoiding the night blindness? The rice itself. The rice itself is having a beta carotene or vitamin A gene. Means what? When the person is taking the normal or the golden rice itself, the person is getting vitamin A. There is no issue of anything related with the night blindness or vision. And here you can see the golden rice, actually which gene is taken? Beta carotene. It is taken from a plant called daffodils. From the plant daffodils, you can see the beta carotene gene is taken, and using a suitable vector, it is transferred into the normal rice or rice sativa to make the golden rice, my dear student. Golden rice is rich in nutrients compared to the normal rice, and that is what we call what? The vitamin A. So what is the answer? The nutritional enhancement is one of the outcome of biotechnology. Cite a suitable example. The example is nothing but golden rice, my dear students. Moving into the next question. Cry proteins, cry proteins in Bacillus thuringiensis, the short form is BT, is something special. What is the speciality of cry proteins? What do you mean by cry? Something related with crying? No, my dear student. What is cry? Cry proteins. Listen, please. So when you are looking at bacteria, if this is the bacteria, look at the board. What is the name of the bacteria? Bacillus thuringiensis. Bacillus thuringiensis. And the name of the bacteria is Bacillus thuringiensis. And here you can see this is B and this is T, that is BT, Bacillus thuringiensis. What is the speciality of that bacteria? And what is the speciality of that bacteria, my dear student? That bacteria is having a special type of gene, my dear student. And the special type of genes are called what? Cry genes. And you can see the speciality of the gene that is called cry gene. Why they're called cry gene? Look at the board, my dear students. You know what is the speciality? And here you can see the cry gene can make a protein called cry protein. The cry stands for crystal. And here you can see, initially you can see a very toxic material is there, and this is the endotoxin. This is the toxin which can destroy the organism, but you know it is covered by, look at the board, my dear student, it is covered by a crystal covering. So that's why, is it active or inactive? It is inactive, because the black color what I used here, and that is what we called what the endotoxin, which is highly poisonous, highly toxic, but it is inactivated by a crystal covering, and that's why it is called what, inactive, right? It is why it is called what, protoxin. Protoxin, inactive toxin, my dear student, and that inactive toxin is called what, protoxin. And here the protoxin, why it is called inactive, why it's called protoxin? Because you can see the toxic material, the endotoxin, is covered by a crystal covering the cry gene, crystal covering the cry protein. Okay, so here you can see what is the speciality, this cry protein. What is the speciality? The cry protein, whenever the in BT cotton, in BT brinjal, BT soybean, BT rice, BT tomato, what we are doing, we are taking the genes. And what are the genes, my dear student? We are taking cry1AC, we are taking cry2AB, we are taking cry1AB. So we are taking different types of cry genes, my dear student. Cry1AC, cry2AB, and cry1AB. And you know, cry1AC and cry2AB for the cotton bollworms, my dear student. And here it is for corn borer. So cry1AC and cry2AB for the cotton bollworms, and cry1AB for corn borers, my dear student. And that is what we called what, the different types of cry genes. Okay, so the cry proteins in BT is something special, because cry genes are so special, and it is not making any harmful effect for the bacteria. It is not making any harmful effect for the host plant, but the problem is for the cotton bollworm. You already studied what will happen when the protoxin, when the cotton plant is infected by, when the BT cotton plant is infected by or ingested by the cotton bollworm, the larvae of the cotton bollworm, what will happen? The protoxin is entering into the alkaline gut. When it enters the alkaline gut, it is the cry protein, the crystal gene, the crystal covering is completely dissolved, you can see, and the protoxin is converted into an activated toxin called endotoxin, and that endotoxin make complete damage to the midgut epithelium. The midgut epithelial cells are undergoing breaking and lysis, swelling, and finally that is leading into the death of the pest, my dear student. That is what we call the pest-resistant plant developed by biotechnology, the recombinant DNA technology. So chance for asking that question, BT products. And you know, in the field of biotechnology, in the field of agriculture biotechnology, there are two pest-resistant plants are given in your textbook. One is called the BT products, and another one is called RNA interference. And you can see the tobacco plant is infected by a particular nematode called Meloidigyne incognita. The Meloidigyne incognita is affecting the root of the tobacco plant and causing the root knot disease. Whenever the root is affected, that will disturb the root, the water absorption mechanism, and that will automatically reduce the rate of photosynthesis, and that will reduce the yield of the plant. And the farmers have no traditional methods to control the pest, because the pest is located under the soil. Manual removal as well as pesticide application, these are the conventional traditional method. They are not applicable in case of a tobacco root knot disease. In that case, you can see what, there is only one solution. It is nothing but the biotechnological solution itself. What is that? And you know, almost all eukaryotic organisms are using one method called what, cellular defense method, using RNA interference, the silencing of mRNA. You already studied DNA can change into mRNA by, no, DNA can change. From DNA, you can see RNA is transcribed, and after the transcription, you can see that mRNA is responsible for coding the protein, the amino acid sequence. And as you all know that here you can see the double-stranded DNA and single-stranded RNA. Single-stranded RNA, we can silence the single-stranded RNA. We can silence the mRNA by making it double-stranded. So that's why what we are doing here, you can using the transposons. What are transposons? The mobile genetic elements, and it is also called the jumping genes. The mobile genetic elements or jumping genes or transposons, or using retroviruses. From the RNA, we are making its complementary DNA. and that complementary DNA is inserted into the tobacco plant, and the tobacco plant can make double-stranded RNA instead of single-stranded RNA. The double-stranded RNA is having a sense strand as well as anti-sense strand, you can see. And when a pest meloidigain incognitia ingest the tobacco root, what will happen? The double-stranded RNA enters the small interfering double-stranded RNA. The small interfering double-stranded RNA enter into the host, enter into the pest, you can see, and it is dividing into sense and antisense, and that antisense strand is complementary to the mRNA of the meloidigain incognitia. Both are combining to make the RNA silenced, to make the process silenced and inactivated. And and after that you studied, right, the biotechnological application in the field of medicine, that is red biotechnology. The red biotechnology, the first one, the therapeutic drugs itself. In the medicine field, one of the most important steps is nothing but the therapy. The gene therapy is there, as well as therapeutic drugs are there. The most common therapeutic drug is nothing but the genetically engineered insulin. Most of the people are suffering due to one problem called diabetes mellitus nowadays. And the diabetes mellitus is always a severe issue. So that's why there is only one solution for diabetes mellitus. It is nothing but the intake of insulin injection, my dear students. So insulin injection means what? There are so many issues are there interrelated with the source of insulin. Initially, you can see the slaughtered cattle and slaughtered pigs are used for the source of human insulin. But you can see, that there are so many side effects, there are so many allergic response, there are so many hypersensitivity issues are there related with that. And finally, you can see the biotechnology, the Eli Lilly company in America. The Eli Lilly company developed a technology to develop the first human insulin, that is what we call the humulin product, my dear student. And here you can see insulin is not produced directly. Human insulin is produced by an artificial method. That is biotechnology, the recombinant DNA technology. But here the question is, what is the difference between pro-insulin and insulin? What is pro-insulin? Look at the board, it is a little bit difficult to understand the topic, but first of all, we can discuss about insulin. Okay, so look at the board, the insulin you can see. Look at the board, please. Insulin, when you are looking the insulin, if this is the insulin, you can see there is a strand is there called A chain. And after that, you can see there is one more strand is there called a B chain. This is called A peptide, and this is called a B peptide. There are two strands. One is called A chain, and other one is called what? B chain. In your textbook, there are two chains are given, but at the same time, you have to study that A peptide is having 21 amino acids, not given in the textbook. And B peptide is having 30 amino acids, my dear student, 30 amino acids. So 21 amino acids and 30 amino acids total. How many amino acids are there in insulin, my dear student? That is 51 amino acids are there in insulin. And here the speciality, this is the first one, second one, third one, fourth one, like that. How many amino acids? Like that, how many amino acids, my dear student? The amino acid, the total number of amino acids, it is 21. I'm not drawing all the 21, but this is the method, amino acid, amino acid, amino acid, you can see. And the number of the amino acid is important. Here you can see that is 30 amino acid. Here you have to understand one thing. Here you can see, and there is a disulfide linkages. What are the disulfide linkages? Here you can see. Look at the board, it is difficult. This is the A chain, right? And this is the 6th position, and this is the 11th position. There is a disulfide linkage between 6 and 11. So the first disulfide linkage between A6 and A11. A6 and A11 means what? 6th amino acid of the A chain and 11th amino acid of the A chain. They are connected by disulfide bond or disulfide bridge. And here you can see this is the 7th one, right? This is the 7th one, this is the 7th one, right? And here you can see the second disulfide bond. So that's why, what is the second one? That is between A7 and B7. A7 means what? The 7th amino acid of the A chain and the 7th amino acid of the B chain. And what is the last one? Here you can see this is the 21. So just before this is the 20th one, and here it is the 19th one, and here you can see this is the 3rd one. So that's why that is between A20, B19. So these are the three these are the three disulfide bond between the amino acids. The first disulfide bond is between only in A chain, and the second and third between A chain and B chain. And this is what we call the real insulin. And what is pro insulin, my dear students? And the same diagram, look at the board please, and the same diagram, there is an A chain and there is a B chain. And here they are all connected by, here you can see a disulfide, here you can see a disulfide, here also you can see a disulfide. And there is an additional, look at the board, this is the B chain, right? This is the A chain, right? There is an additional connection is there, and this is what we call what? C peptide, my dear students. And what is the difference between these two? Here you can see this is the insulin, this is a pro-insulin. Pro-insulin is having an additional C peptide. There is an additional C peptide, my dear students. So that's why the C peptide means what? If C peptide is there, it is a pro-hormone. It is a pro-hormone. In your textbook, pro-hormone, also called what? Pro-insulin. In this case, what do you mean pro-hormone? Inactive. In the previous question, we studied protoxin, right? Inactive. Here, pro-hormone is inactive. Pro-insulin is inactive, my dear students. And here you can see pro-hormone or pro-insulin is inactive. What is the reason behind that inactive nature? The inactive nature is due to the C peptide, the connecting peptide. And that's why, what is that? The C peptide is removed. When you remove the C peptide, the pro-insulin is converting into only two chains, that is called insulin, and that is what? What is the next one? This is the A chain, B chain, disulfide. The C peptide is extra, and this is what we call what? Pro-insulin to insulin. That process is called what? Processing or maturation. Okay, so this is one of the important step in the development of the biotechnological process of human insulin. That is the difference. So pro-insulin and insulin, this is the major difference, my dear students. Moving into the next one. Name the following. There are three sub-questions are there in the single question itself. And what is the first one? Name the following. The first one, biological product for emphysema. You know that after human insulin, there is a topic molecular diagnosis, not given here. Molecular diagnosis means what? What is molecular diagnosis? Generally you can see the diagnosis of a disease is always a Herculean task in front of the doctors. Because you can see it is one of the most important point is it is very difficult to predict the human body. Human body is the most complicated and most complex organ system in the world. We are all having unique human physiology. My body physiology is totally different from your body physiology. The reason is whatever the food I am taking, whatever the genes I am having, whatever the environment I am experiencing, all are totally from you. That is why the reason we are all having different types of human physiology. So for a doctor, you can see identifying or diagnosing the secret behind the disease is always a Herculean task. The doctors are using some common mechanisms, right? What you can see the serum test, the blood test, the urine test, or scanning, or you can see the just examining our body using the stethoscope. But you can see they are all giving the idea. It's very difficult to point out the disease at an earlier stage. A late stage means you can see the disease is diagnosed in a later stage means what? The pathogen completely damaged that particular target organ. Early diagnosis is always good, as well as that prevention is better than cure, right? So prophylactic measures are also very, very important. Prophylactic measures. So here you can see the early diagnosis is mainly done by PCR. That that PCR, the polymerase chain reaction for DNA, and RT-PCR for the RNA. DNA probes are also a special type of mechanism by which probes are single-stranded DNA or RNA. DNA probes as well as RNA probes, and the probes are the single-stranded or RNA, single-stranded DNA or single-stranded RNA, which is labeled with a radioactive material, and that is allowed to hybridize with the normal DNA. If a person is having any mutated DNA is not hybridizing with the DNA probes what we are providing. So that's why what will happen? If it is not hybridizing with the DNA probe, it is not appeared in the photography film. If it is not appeared, the mutated gene is not appeared means what? The doctor can assume that a gene which is located in that particular chromosome, in that particular position is mutated. That indicates the symptom of, or that indicates the presence of a particular disease. That is what we call what the DNA probes and RNA probes. Okay, and there is one more technique, that is antigen-antibody interaction. And whenever an antigen enters into your body, automatically your body is producing antibody. Means what? Antigen-antibody interactions are diagnosed by a type of method called ELISA, ELISA, enzyme linked. Here one question is there, the molecular diagnosis based on, molecular diagnosis means diagnosis at the molecular level, diagnosis at the protein level, diagnosis at the DNA level, diagnosis at the amino acid level. That is called molecular level diagnosis. Earlier stage diagnosis, molecular diagnosis using antigen and antibody interaction, and that method you have to write in capital letter. That is called ELISA method. ELISA, ELISA method, enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay is the technique. That is what we call the molecular diagnosis based on antigen and antibody reaction. And here you can see, and there are so many transgenic animals are there, my dear student. That is the next topic in your textbook. There are so many transgenic animals are there, but more than 95% of the transgenic animals are mice itself. The reason, most of the biotechnological experiments and most of the other experiments, we are using the mice as the major experimental organism. And the transgenic animals are used for studying about the normal physiology and development. You study, in your textbook, the normal physiology and development is studied using the transgenic animals. Insulin-like growth factors, the effect of insulin-like growth factors are also studied using the transgenic animal. The second use is nothing but study of diseases, and different type of study or the pathogen, the disease models are available. And you can see the cystic fibrosis, the what we can say, the Alzheimer's disease. There are different diseases. You can see the even the cancer, different types of cancers are also, this particular disease models are available. So altogether we can say that that is the third one. And the third one is called biological products. And the organisms, the transgenic animals are used for developing the different types of biologically important products, you can see. And there is one of the most common disease among the chronic cigarette smoking people. That is what we call emphysema. Emphysema is one of the lung alveoli damage disorder, and that is due to chronic cigarette smoking. When a person is smoking cigarette for long duration, the lung alveoli get completely damaged, and that is leading into suffocation, and finally that is leading into the death of the person. The disease is called emphysema. And emphysema is treated by a particular medicine called alpha-1 antitrypsin, and that alpha-1 antitrypsin is a product of biotechnology using transgenic animal. So biological product for emphysema, that is nothing but alpha-1 antitrypsin. So alpha-1 antitrypsin is used to treat the disease emphysema. Along with you studied, there is a first transgenic cow named Rosie, and the Rosie can produce a special type of milk which is rich in human alpha lactalbumin. And for a human baby, you can see the human breast milk is the best balanced diet to be given. And you can see after 6 or 7 months, you can see the human breast milk is replaced by the cow's milk. Means what? Cow's milk cannot provide any human lactalbumin. But you can see the Rosie, the first transgenic cow that contains a foreign gene. The foreign gene can make a protein that is human alpha lactalbumin. Human alpha lactalbumin, almost 2.4 gram per liter. That much quantity of human alpha lactalbumin is produced, my dear students. That is good for the babies compared to the normal cow's milk. And even the transgenic animals are also used for chemical safety and vaccine safety. And here the question is biological product for emphysema. And here you can see the ethical issues. I already told, no, my dear students, when we are doing a biotechnological experiments, automatically what will happen? And there may be chance for some issues related with the existence of different organisms in the world. That is why the reason biotechnology in human being is completely banned. And here you can see the use of bioresources. And use of bioresources, some countries are rich in bioresources. For example, a country like India. A country like India, Pakistan, Sri Lanka, Bangladesh, our neighboring countries all together, they are rich in bioresources. But a country like America, A country like Britain, they are not rich in bioresources. So that's why what will happen? Our bioresources are taken by the multinational companies without proper compensation, without proper authorization. They are doing the biotechnological research, and they are taking the bio patent. And after the result of the bio patent, we cannot use that particular our own resources. When we want to do a research in our own bioresources, we have to ask the permission of the other multinational companies. And my dear students, it is not possible to tolerate that one. Even our basmati rice, that, and basmati rice is famous for India. Because and nearly 27 documented varieties of basmati rice is there in India, my dear students. Unfortunately, a multinational company from America, they took the bio patent of basmati rice by hybridizing our basmati combines with a semi-dwarf varieties of USA, and they developed a new variety. They took the bio patent, and after that, we were in trouble, my dear students. So like that, you can see after complete arguments or something, finally we are getting all the bio patent at the same time. There are so many such attempts are done for our turmeric and neem. The scientific name is Asadirachta indica, India, our product. So whenever a person, you can see use of bioresources by multinational companies without proper authorization, without proper compensation and to the country or people concerned, and that is what we call biopiracy. Biopiracy, my dear students. Okay, so biopiracy should be prevented. That is why the reason, our India recently amended the second patent bill, my dear students. Okay, moving into the last question. Name the organization, my dear student. Name the organization that addresses the validity of genetic engineering researches in India. Each and every country is having a particular organization to provide. You can see the validity of the research, and India, you can see that organization is called GEAC. 1 mark MCQ type question. Genetic Engineering Approval Committee. Genetic Engineering Approval Committee is an organization in India, by which they are providing the license to do a research for each and every person in India. So if you want to do a research in a particular biotechnological product, you should get the permission from the GEAC, Genetic Engineering Approval Committee. Only after that, they will give the permission. After that, you can do the research, my dear students. And you, the future scientists, and you, the future doctors, you, the future citizens of India, you should have some basic rights as well as you should have the idea about the basic duties, my dear students. So the biotechnological experiments are always having a merits as well as demerits. There are some positive side at the same time, negative side. And here in the textbook, you can see biotechnology 1 and 2, they're giving only the advantages. Okay, so you have to take each and everything wisely. That is the only thing I have to communicate with you. Okay, my dear students, on the March 19th, you are going to perform your board examination in biology, and my best wishes, and the Excel Education Center, we, the team, biology, the whole department is giving, my dear students, the whole blessings and all best wishes to each and every one of you for performing well in your board examination. Thank you so much for being with me for the last few minutes. Thank you so much.